Hello, everyone, and welcome. My name is Eleonora Chegaray. I am Iktos Marketing Director, and on behalf of the team, I would like to thank you for joining us today for the first session of a three-part series, Makya in Action, Integrating 3D Design, QSAR, and Synthetic Feasibility through GenAI. Before we start the usual housekeeping, the session will be 30 minutes long, and then we will have 10 minutes for Q&A. We will be sharing relevant links in the chat, so please make sure to review. To ask questions, please use the Q&A feature. You can type questions in Japanese. Our colleagues will join to translate the questions in the Q&A session. The webinar is recorded and you will receive the link via email within 24 hours. We will also host the recording in our YouTube channel. Please feel free to share the recording with colleagues. So now I will introduce our speaker for the day, Stephanie Gamboa. She's an application scientist at Iktos, and Stephanie holds a degree in pharmaceutical chemistry and a PhD in theoretical and bioinorganic chemistry with a focus on quantum bioinorganic systems. Stephanie gained industry experience at GlaxoSmithKline before joining Iktos, where she works at the intersection of drug discovery, generative AI, and computational chemistry. Thanks a lot, Stephanie, for joining us today, and the floor is yours.
Absolutely. Thank you very much for introduction, Ele. So first of all, welcome everyone, and thank you very, very much for joining this session. So today we'll walk through an end-to-end design workflow targeting GLP-1R, showcasing how generative AI can drive the de novo design all the way to synthesis planning. So we'll explore how Makya helps translate ideas into chemically realistic synthesis-ready molecules while keeping the design process intuitive and data-driven. So let's take a quick look at what we'll cover today. So we'll start by looking at the GLP-1R receptors, all the critical interactions within the binding site, and how we can take advantage of these interactions in order to decide new analogs. Then we'll move into defining our generation strategy, how to decide what kind of optimizations and which constraints to set. After that, we'll explore chemistry-guided generation settings, detailing how we can control efficiency of exploration while maintaining diversity. And finally, we'll review the generated candidates and wrap up the session with how to prioritize synthesis-ready compounds, connecting molecular design to real-world feasibility. So just to kick off today's session, I just want to make a few comments about Iktos GenAI platform and a high-level explanation on how it actually works. So the first step always when working with our platform is, of course, defining your target product profile. So this target product profile can be anything from the crystal structure of your protein, your ligand into the 3D, your data sets, and machine learning models. It can be also chemical constraints, reactivity, and among others. So once we set up this TTP, now we move into our generative AI, which designs novel chemical structures. But this is an unconstrained generation because the way that we work is that Makya and Spaya, it is grounded in real-world chemistry. So the way that our GenAI works is that it pulls from a library of over 1 million commercially available building blocks and more than 2,000 organic reactions. So in other words, every molecule proposed is by design highly likely to be synthesizable. Then when we generate all these molecules batch by batch, then we enter the optimization loop. So these generated molecules are scored using reinforcement learning based on how well they meet these target product profile criteria. So this scoring system acts like a reward function, guiding the AI to prioritize more promising candidates in every iteration. So the result is a tightly integrating chemistry-aware design cycle that doesn't just generate ideas, it also delivers optimized synthesizable molecules that are aligned with real-world project goals. So if you want to know more about our technology, I invite you to scan this code or to click on the link that we will share in the comments. So you'll be able to see more videos, more benchmarks, our latest webinars, and also some series of in silico experimentation. But let's jump directly into today's session, which is related of designing analogs for GLP-1R. So this use case highlights the kind of problem solvings that you can achieve in Makya when working with challenging targets such as the GLP-1R transmembrane protein. So GLP-1R is a particularly important target in treatments of diseases such as, for example, type 2 diabetes. So let's take a closer look at how the peptide actually interacts within the receptor. So the GLP-1R receptor has two main domains, the extracellular domain and the transmembrane domain. The GLP-1 peptide engages with both, which is crucial because the peptide effectively bridges both domains to stabilize the active conformation of the receptor. So there are several peptide-based GLP-1R agonists already on the market. However, there is a pressing need of developing new small agonists. Currently, several small molecules are in clinical developing, notably danuglipron, delivered by Pfizer, showed a promising activity but was unfortunately discontinued after phase 3. What's notable about this molecule is that it achieves a peptide-like activation to engage key residues deep in the 7 transmembrane region. So this discontinuation is actually an open opportunity to explore new molecules because we already know the mechanism. So we want to design new analogs that somehow can keep the similar activation pattern, but with improved design. So one of the key factors behind danuglipron promising results is its strong overlap with the binding mode of the natural agonist GLP-1. So a crucial aspect of the danuglipron is its ability to form interactions deep within the receptor binding pockets through water bridge contacts with residues buried in the transmembrane region. So these interactions help to stabilize the protein ligand complex, effectively mimicking the key contacts made by the natural agonist GLP-1. So additionally, the oxetane groups plays a key role, enhancing potency by enabling additional hydrogen bonds. So with this in mind, with this molecular understanding, we can now move into the design molecules that reinforce these interactions from the very beginning. So with this in mind, if we want to design new compounds using danuglipron as a reference ligand, it is crucial to retain both carboxylic acid and deoxetane as starting point. So we applied a fragment-based approach to generate novel molecules using these key functional groups as the core elements. So let's move now into the design phase and see how we can translate these structural insights into an AI-driven generation strategy. So our objective is to target specific residues that are known to interact with the reference ligand, such as tryptophan, serine, among others. So these form a stable interaction network anchoring the molecule within the binding pockets. But we are not just here to replicate what exists, we also want to discover what new interactions we can reach within the binding sites, such as residues like leucine 217, allowing us to generate a more diverse set of molecules. So in Makya, we'll then combine 3 components to drive this design. The first one is a fragment growing generation starting from our defined core. The second is a QSAR model that guides optimization towards activity. And the third, a structure-based rewards derived from docking and key protein ligand interaction fingerprints, ensuring that generated molecules maintain these favorable interactions, but we are able to add new ones such as the leucine that I named before. So now what I propose is that we switch to our platform, which is Makya, and I will show you all the modules and everything that you can achieve within the platform. So this is Makya, and before everything, as I said, what we're going to see here is how you can use all the platform and everything just to explore your projects. So everything starts with the project data. So by uploading it, you define the chemical space where the generation will take place, and then you can use it later to train your own predictive models. Then you can also integrate pains and toxicity dictionaries, ensuring that generated molecules stay within safe, relevant boundaries. Then you can also add your docking files to enable 3D guided generations and use also the QSAR models to train machine learning models that can guide the molecule scoring. Next up, also you can use your retrosynthesis preferences to account, for example, for building blocks availability, reaction times, provider, maximum number of steps, etc. Then Makya offers both the 3D ligand-based and also 3D structure-based modules to guide generations through shape similarity, pharmacophore, docking interactions, and protein-ligand interaction fingerprints. Then you can also score molecules independently using combinations of early kits, QSAR, 3D, and APIs-based metrics. And finally, you are ready to launch your generators and explore a new wave of design possibility within Makya. So let's go back into our presentation now that we introduced already our platform. Now let's jump into how Makya operates in general. And so now let's go back into our project with the GLP-1R and look at how we can apply this workflow to design better molecules. So in Makya, structure and information can be directly used as reward function to guide molecule generation. So here you see two rewards we use to optimize the molecules. On the left, we use MDOT docking, where each generated molecule is automatically docked during the run. And on the right, through fingerprint-based interaction analysis, we quantify how well new components reproduce critical ligand-protein contacts. So in this approach, we allowed us, the AI allowed us just to target exactly what kind of interactions are important for you and you want to retain during your generation. So once again, I invite you to jump into Makya and I will show you how it actually works in the software. So if you want to work with your 3D structure base, the first thing that you need to do is to upload your PDV and your STF into the software, which I already did for the purpose of this demo. So once you do that, you will be able to access the platform. And once you access the platform, you will be able to see the molecules within the binding pockets. And for example, here you can explore your ligand within the active site. And in this case, we're going to dock 4 water molecules as well. So before we move forward, just to clarify that with Makya, we use 2 docking softwares, which are Bina and RDoc. So with these 2, you get to decide which one you want to use. And also with Makya, you can work with docking water molecules, cofactors, and also metal ions. So now that we define the reward with respect to the software, now we need to define the rewards with respect to the key interaction fingerprints. So at Iktos, we developed what we call the contact score. That is a proprietary tool that helps us guiding the generations toward these interactions. To do that, we go into the 3D structure base. And here in the 3D structure base, we need to create a new project, which I'm going to do now. And I'm going to name webinar demo. And then here you need to choose which docking software you want to work with. And the one that we've created is with RDoc. So then after that, you can decide which kind of the settings that you want to work with. And when you play and when you click next, you're going to be redirected into the visualization page. And here, very importantly and very cool feature, you will be able to see your ligand, your protein, and also key residues. And with these key residues, the non-covalent interactions between them and your reference ligand. So if we explore these protein pockets, here you see we have the part of the oxetane and the carboxylic acid, which we are not going to touch, and we're going to leave it like this, and we're going to grow into this direction of the protein pockets. So here you can identify more or less an hydrophobic region, and then a region where you have pi stacking and maybe pi cation interactions that it would be interesting just to explore. So as I told you, in Makya, we work with a system of rewarding certain kind of key interaction fingerprints. So to do that, we have a system of weight. This weight, it means that the highest the weight, the most important your interaction is. Like for example, here, I'm going to give a weight of 3 to certain interactions, and I'm going to deactivate other ones. They are not important for the generation because they are not in the part of the protein pockets that it's important for me. And as you can see here, this interaction, these ways, the visualization sphere increased because I gave them a certain ways. Then we're going to do the same, for example, with the pi stacking. Here, this residue 381, here it's also important, so I want to preserve it. And also very importantly, if there are interactions that they are new and they are not detected within Makya, you can easily add them by just adding a new residue. In this case, we're gonna add pi stacking, and the residue that I am interested in is residue 33. And then the atoms that are going to participate in this interaction. And in this case, I'm going to give a weight of 6 because it's a very important residue for me. Then we continue doing the same. I'm going to deactivate the pi cation, the pi hydrophobic as well. And here I'm going to add the interactions with residue, the lysine 217, the one that I mentioned before. And I'm going to add interactions for all of them for 3 different atoms, as you can see here, and then you can add easily and add a ways to all of them just to guide the generation into targeting these 4 interactions. So this is all you need to do into Makya. Then you click save and you are ready to go. And then as you can see here, we have all our contacts, all our interactions just to explore the generation, starting from this fragment and coming into this direction. Okay, so now that we've seen here, let's continue into our next reward, which is a QSAR reward, which is a machine learning model reward that it's also possible to construct within Makya. So with these QSAR models, you will be able to have machine learning models based, for example, on biological activity or ADME properties. And once again, let's have a look at how you can actually work with this within our platform. So go back to Makya, and now if you're working with QSAR models, of course, you need your own data sets. And to do so, we need to go into the data sets. Here you can upload yours in the form of CSV or SDF. And here I've done it already for the purpose of this demo. So let's take a look at how it looks after you upload it. So here you will be able to visualize all the molecules with the different kind of targets. And in this particular case, we're going to be focused in the PEC50. So once you have uploaded your data, now you need to build your QSAR. So to do that, we go into the QSAR models. And here, once again, we need to create a project. So I'm going to call it once again, the demo webinar. And once it's created, you will be able to decide data set that you want to work with. In this particular case, is the PAC50, and then the targets, which is the PEC50, the one that I want to add. So very importantly, in Makya, we work with continuous classification. So we need to tell the software the threshold for where good and bad molecules, or in this case, active or non-active molecules are. So the software is able to train the model. So for example, here, I give a threshold of 8.5, meaning that I have 41 molecules actives and 23 molecules non-actives. Then you can save it, and just like that, you are ready to launch your training. You just need to click run, and then the model will start to be trained. I already prepared one so we can review together the results. But basically, what Makya will give you here is different kind of metrics. Like, for example, we're going to give you the AUC, precision, recall, F1 score. We also give you different kind of plots, like, for example, precision versus recall, and also confusion matrix. So we do this because you as a user will be able to decide if this machine learning model is good enough to be used, or if, on the other hand, you need to retrain the model with different kind of threshold, or maybe adding more data. Okay, so now that we have already set up our 2 rewards, we still have one more thing to decide, and it's like our starting fragment. So now I present you here 2 strategies that we follow in the generation for this case. One is an expansion starting from a piperazine, and the other one is a ring closure. So whenever you are looking for an initial fragment, of course, you have many ways to decide from where to start. start. But in some cases, maybe this initial fragment is not as intuitive, like for example, in the ring closure. So in that way, we also do have another software, which is called SPIA. And SPIA basically complements Makya by assessing the synthetic feasibility of molecules. So it basically evaluates potential synthetic routes and helps identify suitable starting material from commercial providers, and therefore it would help you just to find the initial fragment. So let's switch into SPIA. Here and in Inspire, you will see that it's very simple. The only thing that you need to do is to input, in this case, the smile of my initial, my reference molecule, or you can also draw it. And then when you press start, what will happen is that Spire will find different kind of routes for the disconnection of these molecules. Like for example, here you will see that all the routes, they are arranged based on what we call the R score. So the R score is our proprietary retrosynthesis score, which will give you an indication of how well that molecule can be synthesized or not. So in this case, if you have an R score of 0, it means that the molecule cannot be synthesized through that route. If you have an R score of 1, then now that molecule, it is 100% synthesizable, and it means that the algorithm found a 100% match. So let's explore the second route, for example, here. And so here we will see that this route is only one step. We can explore different kind of references using SPIA. Like for example, here we're going to give you the name reaction, also the pattern from where this process was pulled. And then if possible, we also extract the description. Then if you click here, you will have access to the initial building blocks with the different kind of providers that we have in our catalogs, also the price per gram and delivery time. Then we have this intermediate that is a literature intermediate. So it means that we can continue exploring different kind of routes in order to arrive here. And that's exactly what we do with SPIA. We can ask SPIA, once we identify a molecule, hey, can you expand it? Can you give me more retrosynthesis routes? And that's what happened here. SPIA was able to find exactly what we wanted. We wanted to pursue a ring closure starting from kind of these initial fragments. And so as you can see, you can use our platform not only to find retrosynthesis routes, but also to find suitable initial fragments to grow your molecules. So without any further ado, I'm gonna click, gonna copy the smiles, and I'm gonna go back into Makya to set up our generators. So to do so, we need to go into the generation. And here in the generation, once again, you need to create a new one. So that's what we're gonna do here, demo webinar. And so in Makya, we have 3 algorithms, which are fragment growing, fragment linking, and fine tuning. Since we are growing a molecule, we are going to be using, of course, the fragment growing, and that's the one that I am selecting here. So after that, we are going to be redirected into the generation setups. So heads up here, you will see that we have many settings. We won't be able to go through all of them, but in the upcoming webinars, we're going to explore some of them that we won't have the time to do it today. So the first thing that we need to do is, of course, to set up our chemical constraints. And to do that, we need to go into reaction center. So here we're going to just paste or draw the smiles of your molecule. Then if we go back into SPIA, we have 1 and 2 steps. So we're going to ask the algorithm to at least generate molecules with 2 different generation steps in order to give us our final molecule. And just like this, you would be able to set up and to start growing your generation. But since we like to give control also to the chemist, if you know the kind of reactivity and you know the kind of atoms that are involved in your activity, you're also able to do so using our platform. So here in this way, we are telling the software that these 4 atoms are the one involved in the reactivity, and those are the ones that our software should use to generate molecules. As I said before, this is not mandatory, but this is a very cool tool if you already know what kind of reactions you want to see. So then after that, you can also set up constraints from the point of view of the building blocks. Like here we have different kind of Phys-Chem. So in this case, I'm gonna ask the software to work only with with building block with not more than 3 building blocks. And then you can also work with substructural constraints, whether you wanna see them or you want to forbid them in your building blocks. Then you can do exactly the same with the products. In this case, I'm gonna work with 8 a number of ring as maximum, 11 rotatable bonds, 0.3 as minimum for QED, and 650 for the molecular weight. And then I'm gonna add a paints and talks. Basically, the paints and talks, as I mentioned before, is a list of substructures that you absolutely don't want to see in your generation. And that's what is gonna happen. Makya will generate molecules and then will filter out all the molecules matching this list. So we're set up with the constraints from the chemistry point of view. Now we need to set up the rewards. So in this case, we said that we have QSAR and 3D. So with the QSAR, the only thing that you need to do is to select that you want the one that you want to use. You can use 1, 2, 3, the numbers that you want. In this case, I only want activity. And then we are going to select the 3D structure base, which is the one that we created together. So here we have all the interactions, and this is the setting that we've done together before. And then just to proceed with the generation, you need to create an anchor. And so I'm going to use the one that is included in my initial fragment. And just like that, you are ready to start your generation and to wait for Makya to give you results. So if you come here, I just want to show you how to start the generator, but it's very simple. You just need to press run, and that would be it. So I already prepared the results just so we can explore how it looks, the generation results, and how you can expect after the run has finished or you have stopped it. So basically, we're going to show you a listing of molecules, and these molecules, they are going to be sorted out with different kind of metrics. In this case, it's Iktos ranking, but it could be docking, contact score, prediction. Then you can use also these to filter molecules based on different kind of thresholds that you want to see, and just reduce the visualization of your molecules. Like for example, here I went for around 7,000 to 800. So this way you can explore further your results. Personally, I selected different kind of results because of the way that they fit with the binding pocket and the way that the characteristics that they had. And I'm going to show you the result of one of them. So this molecule, for me, was a very good one because if you observe it in the docking post, which is also another thing that you can do with app software, you can not only generate your molecules, but you can visualize it in 3D, docked inside of the protein pocket with all the interactions. And so in this case, this molecule is very interesting, and it was giving me the new interactions that I was looking for. So that's one of the reasons why I selected, and of course, because of the chemistry point of view. Then with this molecule, you can also send it into SPIA and explore different kind of retrosynthesis routes. And so if you click here, then you're going to be redirected into SPIA. And here, once again, you have different kind of routes and different kind of disconnections. Like for example, here, SPIA was able to find a very interesting retrosynthesis route, exploring different kind of building blocks, different steps, and different kind of references. So, and now that we've gone through the rewards, the generations, the results, let's go back into the final step of this webinar. And so now that we have the generation is completed, let's analyze the results and how we identify the most promising candidates. So you will see here that we selected different kind of molecules. We did this, since this is a first run of generator, we just focus on scaffold, non-decoration, just to see then the ideas that Makya was giving us. So this is coming from the p-pyrrazine starting fragment, and this is coming from the ring closure experiments. So for all of these, of course, we've identified, for example, geminal linkers, where substitution increases local rigidity and may improve the receptor alignment. And the second focus, for example, in the linker flexibility, exploring variants that could accommodate better the conformational changes. But the important message here is that these follow-up compounds are excellent starting points and served an inspiration for manual curated list for you as a chemist to target further exploration. Like for example, here, I just wanted to show you a list that we created of molecules, follow-up molecules from what Makya was suggested. So this step illustrates how AI-driven design complement chemistry intuition. We don't want just to substitute our knowledge. We just want to, it's just kind of a tool for the chemist to generate diversity, and then from there to move forward with different kind of propositions. So just to finish, I just wanted to show you this video. This is our reference ligand, the danulipron, together with non-covalent interactions. And here I'm going to show you two analogs that Makya was able to generate. So this is a perfect example of Makya's ability to explore new chemical ideas without losing essential contacts and enforcing the generation to explore new ones. Like for example, tryptophan 33 and lysine 217, which were the new interactions. And then in the second one, and the second compound, you see that it's shifting deeper into the cavity thanks to the two linkers, but also it's keeping like the new interactions that we wanted to explore. So altogether, what we wanted to show you with this video is that basically both poses overlay remarkably well with the reference, showing how Makya's AI-driven generation not only diversifies structure, but also preserves the 3D structure-based relevance that you as a user wants to see in your generated molecules. So just to finish our webinar for today, we went through all the workflow since just with the problematic, identifying the starting point, identifying the rewards, and then picking our molecules. Now the question is, how can I now explore how these molecules, how I'm going to actually make these molecules in the lab? So you had already an idea of Spaya, which is our retrosynthesis AI, but basically with Spaya, you will be able to explore many ways of finding retrosynthesis routes or synthetic ways of synthesizing your molecules. Like for example, here you will see that with Spaya, you will be able just to ask the software the retrosynthesis analysis with a maximum number of steps, with a maximum parts per gram, delivery day. If there are certain name reactions that you want to explore or you want to forbid, it's also possible to do so. If you're working with intermediates, like in this case, our ring closure intermediates, then it's also possible just to find routes using Spaya. Then Spaya will generate different kind of routes, and you will be able just to define it. So just to give you a heads up on how you can work, you can prioritize on route depth. For example, if you're in early stages of your project where you don't want to waste a lot of time with chemistry, and then you will prioritize routes that are more simpler than the others. You can also prioritize on price and availability of starting materials. Like for example, here you have different kind of prices, but different kind of delivery times as well. Then also you can prioritize on required chemistry because you will be able to explore the references. For example, you have condensation and alkylation in this case, and so you can decide which kind of reaction fits the best with the kind of chemistry that you are doing. And so with this, that's the end of our today's webinar. And just a conclusion and just a last word, what I wanted to say is that at Iktos, we believe that AI is at the service of the chemist. So the idea of Makya, Spaya, and Iktos platform is just to give you the power to the chemist and just to simplify your work. But always, what we want to do is to take in consideration the chemist. Yeah, that's all for today. I will give back the word to Eleonora. And thank you very much.
Thank you, Stephanie, very much for the presentation. presentation, really lively, dynamic. Everybody, you will have access to the recording if you want to go through any of the sections that Stephanie presented. And also please check the chat because we have the links to our YouTube channel where you will find more training videos on Makya and Spaya. So now for the Q&A session, we're also welcoming, in addition to Stephanie, welcoming Victor Cachou and Misugi Ebihara. They are both our application scientists in Asia and Japan, and they will be joining the session to answer your questions. You also have their contact details in the chat if you want to reach out directly for a question on how to use Makya. So now I will read the first question that we have. Let me go ahead. Even if the training data is limited, can the prediction model still be applicable to the generated compounds that are considered to be in a larger chemical space?
Yeah, of course, I can take one. And in Makya, you are able to create machine learning models. Of course, data scientist here, Victoire, she will correct me with at least 35 data points. So even if your data set is limited, you will be able to create models in Makya, no problem with that. Then it's a question on the kind of data that you're using to create your machine learning models. But I will let Victoire as well because she's
Yes.
the expert in the data science, just to complement the question.
Yes, no problem. The question is a very interesting one because basically the question is uh, how do you make sure when you train models on limited set of data that they are still applicable to the larger chemical space that you are generating, especially that we're generating with high diversity. To give tools to the users to make sure that they are in their applicability domain of their models, we created a confidence score, which is not exactly similar to the Tanimoto similarity, but it's usually quite correlated with it. But basically, the confidence scores measures whether the substructures present in the generated molecules are found as well in your training data set. So for example, if you generate a molecule that has sulfonamide and you have 0 sulfonamide in your training set, your confidence will be low because we have no way of knowing what a sulfonamide can have as an impact on the property you're trying to predict. So these are tools that we introduce that can help you make sure that you are in your applicability domain.
Thank you
And as I
very
show
much.
you here, sorry, and as
Oh,
I show you here, you still see it, my screen, right? And as I show
yes,
you
really.
here, this is the confidence score that we give you during the generation.
Thanks very much, Stephanie and Victor, for the answer. We will move now to our next question. How good the demonstrated use case will be if there is no protein ligand complex information, only tool reference molecule?
Yeah, of course. So we will explore this in upcoming webinars as well. But as I said before, in Makya, you can also, if you're not structurally enabled, you also have the possibility to work with 2D or with 3D ligand base, in where we're going to compute the pharmacophore similarity and the 3D shape similarity of your molecule, your reference molecule. So it would work in the same way, but in this case without the protein. So it's absolutely possible to do it as well.
Thanks, Stephanie. And also really good to mention, please remember that in 2 more weeks, we will have another session with the next use case.
Yeah, and we'll explore there exactly in that webinar the how to use the 3D ligand base. Yeah.
Super. Thanks. So we move to the next question. Could in-house machine learning or QSAR models be plugged into Iktos platform?
Yes, absolutely. We do have a connection through an API gateway, which is very secure, and you can connect your in-house models into the platform without any problem. And you can use them after to score your molecules. Yeah.
Thank you, Stephanie. So we have another question from Saikan. Thank you very much for the interest. Can we calculate synthetic accessibility score for the generated molecule?
I'm sorry, before? What? I didn't.
Can we calculate synthetic accessibility score for the generated molecule?
Yes,
So maybe you can go
absolutely.
a bit
Yeah, yeah,
further
yeah.
on
Yeah,
how
yeah,
it.
absolutely. It is possible to calculate the retrosynthesis. We will show it as well in another webinar. But let's stop the generator first because this is the one that I launched. But basically, we have the retrosynthesis option here. So I will show you a bit more in detail. But with this retrosynthesis option, you will be able to input different kind of settings. Like for example, as I showed before, intermediates, price per gram, price, delivery time, you name it. And then Makya will connect to Spaya, and Spaya will find routes and will give you the R score of the generated molecules. Yes.
Thank you very much. So we have 1 more minute. I'll go through some of our last questions. You mentioned synthesis-aware design with name reactions. Can we restrict the model to a company's internal reaction set or building block library?
Yes, it is possible to do so. In Makya, you can integrate, we can retrain or integrate your in-house building blocks and reactions. And so the software will generate only using that information. Yeah.
Thank you very much. One more question. Is it possible to generate compounds and optimize them, also taking into account ADME properties of these molecules? Example, metabolic stability, permeability, etc.
Yeah, it is possible. We do have, I mean, if you have your data, you can create your machine learning models within the software in Makya, as we did it today, and you can use them, and you can use as many as you want in the generation. And then we also have global models, like generic models in our platform that they can also be used as a post-processing score. And yeah, it is absolutely possible.
Thank you very much. Now we have a question around data. So maybe Victoire, you can comfort this one. Would
Yeah.
the first question is, will you be going to showcase any demo where the spread of the IC50 is very narrow? I mean, what would be the success rate of such a use case in your platform?
If your PIC50 spread is very narrow, it's going to be more difficult to train models, of course, because we have less margin to know what is a good molecule and what is a bad molecule. So it's hard to answer this question in a general matter. It will really depend on your data. And so without looking at your data, I cannot say for sure that the model will succeed or fail at predicting on larger compounds. But of course, it's going to be more complicated. In that case, it can be interesting to complement your generation, your analysis, by using more than the PIC50, by using additional scores and metrics that are important for you to generate compounds with a more diverse range of possible properties.
Thanks a lot, Victoire. I will mention now that we are on time, but we will keep going as questions are coming because we're happy to have the opportunity to engage with our audience. So we have a following question, and maybe you can expand a little bit on the data capabilities. He's mentioning that he missed the lecture, so please access the recording. Yet he would like to know how Makya is different from other admit lab tools. So other software that could do admit prediction.
I can take that one. So I haven't used myself Admit Lab specifically. So it's not a tool that I've used recently. So it's more complicated to answer precisely. What I can tell you is that Makya, our platform, allows you to train QSAR models on your data. These models are continuous classification, so they're based on classification, but fit better the data around your threshold. We're getting into technical territory, but so basically you can train your own classification models on your own data in Makya, model whose hyperparameters are optimized. And then we also have access to some global models. So if you don't have any data, we have trained our own global ADME models on some public data, on PPB, on clearance, on ERG toxicity. And these are also tools that are available in Makya for scoring compounds and helping you find interesting compounds for your projects.
Thank you very much, Victoire. Maybe we're reaching our last question. How adaptable is the reward function system to different therapeutic modalities? Let's say kinase inhibitors versus ion channel ligands, etc.
Well, the reward function, it's like it's very it's very user friendly, like it's user defined. So if you're working as I show it, if you're working with a protein, you are gonna define which reward you want to use. Like, because the rewards that we are working in our platform, it's very straightforward. It's docking, and so you're gonna use the software that you want to with which you want to use dock your molecules. And then, for example, the contact score, which is that's very, very user defined, because you're gonna guide the algorithm just to generate only molecules, or just to optimize molecules targeting certain interactions. Then, if you're using QSAR, it's highly like customizable, because it's gonna be based on your data. So it depends on the data that you have. And then, with respect to the 3D ligand base, this is also something that we're gonna implement very soon. But we have pharmacophore customizable, where you will be able to customize the pharmacophore of your 3D ligand base. So everything is custom defined here. So it's not just the black box reward that you put your TTP and then you see something coming out. No, everything is guided by you and independently of the target. Yeah.
Thank you very much, Stephanie. So with this, we close today's session. So I want to thank our presenter, Stephanie, and our application scientist, Victoire and Misuki, for joining us today. And of course, all of you. We hope to see you in our next webinar 2 weeks from now to discuss more use cases with Makya. And in the meantime, please reach out